Thanks a lot, Emma. Can you hear me properly? So, skip to the first slide. So, I'd just like to start off firstly just by telling everybody about some good news stories to do with seed-based restoration. And there's a reason for this. So, if we look around parts of Australia and also over in the US and other parts of the world, we do have some good news stories with regards to seed-based restoration. The reason why I'm putting these up here is that you can start to see some of the scales at which seed-based restoration is actually tackling restoration. So in South Australia, is there a little, this one? So in South Australia, in this instance, they actually use some pretty innovative techniques that terrestrial restoration uses, which is removing the topsoil, which has lots of dormant seeds stored in those sediments. And they use them and transplanted that topsoil. They're able to transplant over 700 tonnes of topsoil over a period of 2 years and covered an area of about 61 hectares in this instance. In the US, with Zostra marina, over a 10-year period, they reseeded or broadcast seeded over 71 million seeds into the Chesapeake Bay region. They reseeded 200 hectares, and this area actually, this reseeded area, increased in size to over 2,500 hectares. Again, in South Australia, they have another innovative technique where they facilitated the recruitment of amphibolous seedlings, which have a little grappling hook, which is attached to these large hessian bags. In this instance, they were able to concentrate large numbers of these seedlings if they were placed at the right location and the right time of year. So the work that I'll be looking at today is on a species in Australia, Posidonia australis, which I've been working on for a number of years. I've also been working on Halophila ovalis, but I won't go into that particular species today. But the methods that we're using for Posidonia australis have been directly applicable to the other species. And the reason why I'm not going into Halophila ovalis today is just the maturity of that work is just not quite there yet. So I want to focus on Posidonia australis. So our approach to our research is basically using a systems approach. So if we take the seagrass life cycle here as basically a process, what we're actually trying to do is to identify where there are bottlenecks in the process that are slowing down or preventing seeds from getting to one stage to the next, or even just the availability of seeds in this instance. And then from there, can we actually overcome these bottlenecks? So can we employ some strategies or technologies to get a seed from one that we put out into the field to something that's actually growing into an established plant? And the reason why we actually want to do this is to increase the efficiency. So we can retest any of our overcoming bottlenecks technologies to try to increase those efficiencies, and we can test that back in the system. Whoops. So we just recently completed this research here, and this is in Coburn Sound, which is in Western Australia, just nearby to Perth. And in this particular program, we had 8 locations spread across an environmental gradient. So the environmental gradient is, in this particular northwestern region, is high wave energy. As you move down to the south and to the east side, it's less wave exposure into these environments. We also had other gradients of depth that were dotted across these different locations, and also benthic fauna. And although we couldn't change the benthic fauna, what we did was we added exclusion cages in this instance. We had 8 locations. We had 3 sites within each location, and within each site we had 9 plots set up, and each plot was 1 square metre. We planted 100 seeds into these 1 square metre plots. In total, we hand planted about 22,000 seedlings in this instance. Just to make you aware, this wasn't a restoration exercise. This was mostly an experimental exercise, just trying to identify where there were bottlenecks in the process. So this is a Posidonia australis seedling life cycle. They have a direct developing seedling, so we don't have dormancy as Emma was talking about before. And they go through a number of stages based on the basically utilisation of the starch reserves inside the seed until they become an established seedling and then a juvenile and adult before they're able to reproduce again. And so this is a bit of a complex diagram, but I'll try to explain it. So each one of these boxes is a survival plot for the whole location. This is over time, so time of planting, 1 month, 2.5 months, 4.5, 6.5, and then 1.2 years after planting. These are the more exposed sites to the north, and these are the least exposed sites, or wave exposed sites to the south, and these are our caging enclosures. I'm not going to talk about the caging enclosures today because we had some problems. But what I want to point out in this particular figure is that in these low exposure sites, we got almost 100% loss within the first month. Whereas in these more high exposed sites, we didn't get much loss. We did have some loss. We had quite a lot of survival over the first few months up until winter, okay? Overall, we got 0.07% survivorship for the 22,000 seedlings. So we were quite disappointed by that result, but that wasn't the point of the exercise. We wanted to understand what was actually going on. Always take a positive out of this work. And so what we did is we conducted a sensitivity analysis, and basically what we wanted to understand was where these bottlenecks were occurring or what life history stage. And so in these low exposure sites, we see that the numbers in bold are indicating that it's significant. We're seeing that this first developmental stage is the most sensitive, so in the first month. Whereas in these more high exposure sites, we're seeing that this later development stage is where we're starting to get these large losses of seedlings. And this occurs around the winter months. So why is this actually occurring? What's actually linking up with these major losses? So in locations that are less energetic, we're seeing a lot more benthic fauna in these particular locations. So we had a PhD student from VIMS come over and look at how these animals were interacting with the seeds. So we put the seeds out in the field in these little raceways we devised, and we set up seeds in front of the animals. That's a sand dollar and a sea star. And we looked at how these animals interacted with these seeds and how far they actually moved those seeds. So the distance they actually moved the seeds was over a period of about 10 minutes. So some of these seeds were being moved many centimetres within a few minutes. But the main message here is basically that they were disturbing these seeds, regardless whether they move them a centimetre or 10 centimetres. They were disturbing them in some way. And what we found was that sand dollars in particular were very good at excavating and moving these sediments. And they could basically disturb 1 square metre of sediment every 1 day and up to 8 days. But if you're a seedling, it doesn't matter if you're getting disturbed once in that time or a couple of times. You're basically getting uplifted and pushed somewhere else. And if this happens continuously over time, you're unlikely to survive. The other process that we saw around the winter time was winter storms in Western Australia. So on this side just indicates the number of, or the storms by year. Down here is the year that we did our experiment. So you can see there's a number of storms arising. Whereas on this side, we've got the number of storms arising from the north. So this is a very exposed location to the north. In our particular year, we had a number of storms occurring in just within a month at our particular location. However, if you look back in time, a few years, you see there are times when we see these more benign conditions, so less storminess in the system. So these are potential windows where you might potentially get more recruitment in this particular area. The other bottleneck that we found in our work, we had very low survival, 0.07%. If we're only planting out 100 seeds per square metre, we find it's very difficult to detect that loss. And so what we wanted to do is basically look at whether we could, were we planting enough seeds, and if we increase that number, what would that actually look like? So we modelled this, and obviously it's quite proportional, so we did see some increase in the number of seeds that we'd expect to survive here. So the next step for us was to try to test this and look at ways of actually increasing the number of seeds that we could collect for a restoration program. So we consulted with our terrestrial counterparts who have a lot of expertise in this area. And as Emma mentioned, there's a process that they go through to try to increase the numbers of seeds that they use in restoration, and also the quality of those seeds, how they would actually store them, and any processes they'd actually put them through before they put them out into the field. The great thing about Posidonia australis is that we don't have to worry about dormancy. Once it's released from the fruit, you can see whether it's viable or not. Unless it's got damage or it's a very small seed, they tend to be very healthy. We're getting about 98% viability of those seeds. And so the big issue for us was just collecting large enough quantities to use in restoration trials, and then how do we actually get these back out to the field in a timely enough manner. So I have a little video here just to go through that process. I'll see if I can keep up with it. So this is Posidonia australis fruit here, little yellow things on top of the meadow. So this is Renee here collecting the fruit. So we devised these purpose-built nets to collect the fruit. They're positively buoyant, they float up to the net. A single diver can collect anywhere between 10 to 20,000 fruit within an hour. We take these fruit back to the lab And this is all within about a 24-hour period. Each one of these 500-litre tanks can hold around 25 to 30,000 fruit. The fruit split open, the seeds are negatively buoyant. We're basically getting pure seeds dropping out onto the bottom of the tanks. And so we can take those seeds, we count them based on basically doing a volumetric displacement analysis and getting fairly accurate numbers of seeds. And then we go out and we scatter seeds by hand, either through divers, taking them straight down to the bottom, more sort of precision seeding, if you will. And you can see here they're quite negatively buoyant. They do settle quite quickly to the bottom. And the other way that we do this is via broadcast seeding. So we take the seeds in a bucket, sit off the boat. In this instance, we're here at Rottnest Island on a mooring, and we just throw the seeds over the side. And you can see here them settling quite rapidly to the bottom. And they do have this very nice coverage across the sea floor. So it's quite an efficient way to cover a fairly large area. That took us about 10 minutes to just throw seeds in a 50 square metre area. So these are seedlings that have survived. This is about a 1-year-old seedlings, and this is about 10% survival. So we were reseeding at a rate of about 200 seeds per square metre, or about 5,000 seeds per 25 square metres. And we got 10% survival. And these are 2.5-year-old seedlings that have actually increased in size and have basically starting to produce new shoots. So just to go through some data here, I'll go through this quickly. So these are the sites that we used to do this reseeding work with the high numbers of seeds, just in Cockburn Sound there. And we reseeded at 5,000 seeds per 25 square metres. And basically what you see here is that the best sites are 10% survival. We've got about 15 plants per square metre in that particular location. Other locations we've got 5, 2, and there's obviously some problem locations with 0.5 seeds per square metre. There's a lot of biological activity in this particular location. So we are getting some quite positive results. And what we've found that even now, I haven't included the data here, that basically once they get past this 1-year stage, they tend to persist. So these seedlings are starting to produce new shoots, and they're basically in the environment, and they're quite well established. So just in conclusion, we see that seeds are fundamental to broad-scale restoration. There's a number of studies in Australia and globally that are actually showing this. We're trying to continue to develop the seed restoration, seagrass restoration seed palette by increasing the different species that we're working with, how we collect them, how we treat them, as Emma's working with as well. And we're starting to see some good positive results from that research. We're also finding that recruitment failure is going to continue to be common unless we try to understand the failures. There is always, there's the stigma that seeds are not useful because there's low recruitment. It's low recruitment because we don't really understand why there's low recruitment, and we're not actually investing in the technology and the strategies to try to overcome these problems with low recruitment. And we've tried to implement our research within a framework which can be transferable to different species. So within this sort of process-based approach, we can retest any different strategies that we have to try to improve the efficiencies of restoring seagrasses via seed. Thank you.